Okay, perfect. Welcome everyone to today's seminar hosted by the Berkeley Pharma Tech Foundation. My name is Tiffany Terry. I will be facilitating today's seminar. Our mission is to increase access for scientists and entrepreneurs in biotech through education, mentorship, and professional opportunities. Through our speaker series, industry leaders share insights on cutting edge biotech innovation, career development, and entrepreneurship. Our goal is to foster a community where scientists and builders learn from real experiences and grow together. To get involved, feel free to follow us on LinkedIn for updates. Stay tuned for our upcoming seminars and conferences, and feel free to reach out to Van Lowen, who is the president of the foundation, to explore more ways to engage with our mission. And so today, we are thrilled to welcome our speaker, Will Alaynick. Will is the founder and managing partner of Phase 2 Ventures and the author of Venture Capital for Life Scientists. He is an experienced entrepreneur and investor in life science tools. He has built and scaled multiple companies from academic concept to global commercialization, raising capital from leading venture, private equity, and strategic investors, as well as securing NIH SBIR funding. Prior to his work in venture, Will spent 14 years in academic research and contributed to 33 peer-reviewed publications and 19 patents. Today's session will be interactive as Will is welcoming of questions throughout his presentation. We're just going to ask that you please use the raise hand function. We are excited to learn from his journey and insights. Please welcome me in joining Will.
Great. Thanks, Tiffany. So everybody, Will Alaynick here. I thought I would just step through kind of the history of how I got involved in all of this. And as Tiffany said, I'm happy to answer questions as we go, or if you have questions you want to ask at the end, that's fine too. So, get my slides to work here. There we go. So just as an overview of what I'll talk about today, I'll briefly step through my background and some of the companies I founded, tell you a little bit of the lessons I learned from those experiences and how I made it into starting a venture capital firm. And then as part of that, I wrote the book Venture Capital for Life Scientists as a bit of a study exercise. And then I'll kind of share some takeaways I've gotten over the years. And then if there's still questions at the end, happy to answer those. So this is how I got involved in, this is my first career choice was to try to be a rock star in Seattle. I jokingly say I was the only guy in Seattle who didn't get signed in the 90s. So this is the Pearl Jam show, I think in 93, what they call Drop in the Park. And Eddie Vedder had just fallen out of the rigging onto the stage. And I'm over there on the side there. I was standing next to Dave Grohl briefly, but this was as close as I got to rock star. It was close, but not quite on the stage, just a little bit to the side. So after this, I thought, well, my parents are MDs. That seems like a cool career path. Why don't I get serious and go back to school and see how that goes? So I went to the University of Washington and kind of a pre-med track. And so my academic training kind of started in an HHMI lab with a guy named Richard Palmiter. He was one of the first developers of transgenic mice, which I had never heard of. I'd never heard of the Howard Hughes Medical Institute. And I was just lucky that I got an opportunity to join his lab. And through that, I got introduced to one of his collaborators, a guy named Ron Evans at the Salk Institute, another mouse genetics kind of guy. So I did a PhD in characterizing a knockout mouse of one of the nuclear hormone receptors, a protein called estrogen-related receptor gamma, which is similar to the estrogen receptor, but it doesn't bind any hormones. And then I stayed on as a postdoc in the lab next door. Turns out during my investigations of this mouse, some of the proteins expressed in the spinal cord in what are called gamma motor neurons, as it turns out. And so I worked for another 5.5 years on spinal cord development. Um, you can kind of see on the wall behind me, there's a cell snapshot paper of, how do you build a spinal cord from transcriptional perspective. Um, and then about during that time in my postdoc, I started getting a little bit entrepreneurial. So I found some allied people at UCSD that were also interested in trying to go into business, and we started essentially a little consulting club. And having put our name out there, we got the attention of a professor at UCSD who had a microfluidic cell sorter project, this little chip. And we thought, how hard can this be? You just make a cell sorter. But turns out it was a little harder than we thought. Um, but started that in 2009, and I'll tell you more about that in a moment. Um, and then shortly thereafter, we got introduced to a guy named Bing Ren at UCSD, who was using a technique called Hi-C, which allows you to study the conformation of chromatin. And that also allows you to impute long range information. So if you can determine that, you can use regular Illumina sequencing to do chromosome spanning contigs and figure out phasing. It's really, you have 46 chromosomes, it's not 23 in me, right? It's 46. So that allows you to do also cool things around cancer diagnosis. And then in 2019, we spun out a technology from Northwestern that is using animal-free minimal media for stem cells. And then in 2023, my longtime business partner, Jose Marachis, we've working together since 2007. We thought, we've learned a lot from all of these interactions with the people that invested in us and as operators. It seems like the next phase two, should be to start our own venture fund and take some of the lessons we've learned to try to mentor and support people early in their entrepreneurial careers. And so we currently have 10 investments we've made, and probably for Tiffany's sake, Anson is in Georgia, so that's one of the key geographies. The rest of them are here in San Diego. So a little bit more information about NanoSelect. We found it in 2009. Like I said, it was the technology in Yuwa's lab was a microfluidic chip. This is where you take semiconductor manufacturing techniques to make a mold for PDMS, this silicone rubber, and you can peel that off and stick it on a glass slide, and it'll give you these little channels. And with a simple little piezoelectric disc, these little beepers that you find in things, you can just use that to push and pull the fluid stream left and right, and that's how you do the sorting. We thought, how hard can that be? But what we didn't realize is that you have to have mechanical engineers, fluidic engineers, electronics engineers, optical engineers, software and firmware engineers, sales, support. We basically had our MBA on the streets trying to figure out how to get this company going. And so we wrote a lot of SBIRs to do that. So Yuwa taught us how to get these SBIRs going. For the people who don't know about cell sorting, I call it the coin star for cells. That's how we would explain it to people who aren't from a cell sorting background. And it turns out that our system was slow, but it was also very gentle. So it had a nice application in doing single cell sorting for single cell omics. You don't beat up the cells too much. Or if you're doing single cell cloning, you have high viability of the cells that you've sorted out. And so we built a second generation machine, and even we raised $13.5 million from SBIR awards to do both the initial first machine and then a third generation machine, um, along with 90 million in venture. And that allowed us to get about 450 machines placed out there in the wild. And this third generation machine actually does imaging of the cells. So here's a graphic of that. And what you're seeing here is a cell flowing through the detection zone and alternating blue and yellow lasers scanning it like you would in a scanning confocal. And so it just scans across and the cell goes by and you raster the image from it as it goes by. So the cells are going by at up to 200 cells per second, and those lasers are going back and forth at about 400,000 cycles per second. So it's a pretty fiddly device to build, but we've built it and sold it. Um, and so it was pretty fun to help design and build and think about what the market application would be for that machine. So that's kind of NanoSelect. Any questions about NanoSelect from anybody, or happy to save your questions to the end if that's easier. So the other company I spent quite a bit of time with is Arima Genomics. And as I hinted earlier, I call this 46 in me instead of 23 in me because it allows you to get, with regular Illumina sequencing, you can resolve all 46 chromosomes separate from each other. And with that, it means that if you have a breakpoint, so we used quite a bit of SBIR awards for this as well, and quite a bit of venture investment. The cool thing about this is if you're able to map with high resolution or high certainty where those chromosomes are, if there's a break and you have a fusion event, like the Philadelphia chromosomes, the classic cancer driver, um, you can detect that with high sensitivity at standard sequencing depth. So a few years ago, the company launched what's called the Aventa Fusion Plus for 361 of the most common fusion genes that are known to be drivers of cancer. And then more recently, another test that's tuned towards detection of lymphoma. And so it's kind of just coming across an inflection point from being a research use only application to a clinical diagnostic, which is a pretty major shift in the operation and financing, and just the whole business model is very different. Defined Bioscience is a company we started with IP out of Northwestern and some IP from UCSD to kind of help with the boring stuff of cell culture, right? When we're doing all this work with NanoSelect, we work with a lot of single cells, and oftentimes the sorter or the sorting results were only as good as the cell culture media. And a lot of people just have trouble coming up with easy to use, consistent cell culture media. In addition, people want to get away from using um, animal products. So I jokingly call this vegan cell culture media because there's no animal products in it, right? And this is important because bovine sourced media, fetal calf serum and things like this, oftentimes those animals are ill and they're collected in, it's just not a pretty process and it has a lot of variability. And so you have lot to lot variability on your FBS. And if you're going to ever have those cells go into people, the FDA doesn't want you to have potential exposure to prions or whatever viruses. It's just messy. And so if we can come up with a more defined version of that using recombinant growth factors, it's better for everyone. So this has been funded with a handful of awards as well, and a little bit of venture capital investment. And we've launched, I think, in total 9 products so far, and we're moving towards CGMP manufacture. And in this image, you can see one of our collaborating companies called Novaron is studying how to make iPSCs into neurons and use those neurons to study the spread of tau, for instance, in an Alzheimer's model. And so along the way, one of the things I learned about was being involved in sales, both in the US and setting up distributors internationally. In NanoSelect, we had a presence in about 21 countries by the time I left. in 2024. So I got to do a little bit of travel in the late 2019 and early 2020. I found myself in China in January of 2020, which was interesting to watch, the one English-speaking news channel talk about flu-like illness in Wuhan. Um, and then I even went to Europe after that before things got shut down. But um, then I had to basically learn how to carry out global sales on Zoom, just like everybody else had to figure it out, right? So we had to make a ton of digital assets and videos and do a lot of webinars and just a lot of time on Zoom talking to people and educating them on how to find their customers, how to overcome their potential objections to the product, and educate them on what the product can offer. Make sure we're not telling them something we can't handle. So that was quite a learning experience to work internationally in those times. So with all these experiences, Phase 2 Ventures, we launched this about 3 years ago, and we were targeting life science tools, reagents, largely because that's what we had experience in. The Fund 1 was initiated in May of 2024, and we've made 10 investments so far. And we'd like to argue that our edge is that we're scientists by training. We've been founders of companies. We've operated the companies. And so we have a good understanding of, here's what I intend to do, and here's what actually happens, right? The life kind of jumps up and bites you as you're trying to get things going on a nice, smooth, easy path, right? And so we have advice that we can provide from, in theory, here's what's supposed to happen. And in practice, this is probably closer to what's going to really happen. We're going to launch a second fund later this year. And as I said, we have 10 investments so far. I'm missing a couple of logos here, but if you go to the website, we have more information about our companies. Um, Real briefly, Ribodyne is looking for new markers for immuno-oncology and autoimmune disease. So they have a way of reliably detecting peptides that are previously unappreciated. Xthymic is they want to make cell therapy in a machine that you would find in like a major academic hospital. So you would do your um, uh, plasmapheresis or what's the word I'm looking for? Collect your white cells, and then rather than freeze those down and ship them across the country, they would go down into the basement. You'd have a machine in a nice clean area run by qualified technicians, and you would, in a few days, have your cell product completed, and it would come back upstairs. You don't have to freeze it and ship it, which is a major, you lose a lot of cells and it costs you a lot of time. Lynx Bio is doing microfluidic based arenas where you can take immune cells and cancer cells and basically watch them fight over 4 days. And you can also add and remove small amounts of material for analysis. Anson Biotech does cleanup of antibodies upstream of mass spec. So your cleanup step is really fast. It's just in line. So you can shoot and scoot, I think is what they say when you're doing mass spec quickly. Nevega Therapeutics is using epigenetic mechanisms to downregulate the expression of pain-associated sodium channels to get rid of neuropathic pain while preserving normal pain sensation. Potato is a AI platform that creates scientific protocols for wet lab experiments and dry lab experiments, and even a little bit of automation using plain language interface. And they have a relationship with major publishers to get access to a lot of protocols without running into IP issues. And Morphos Bioscience is a studio investment, meaning a company that we founded at the fund. And this is looking at spatial biology. So this is basically a specialized cover slip you can put on a slide, and it allows you to use sequencing to determine what's in that sample and generate a spatial image from sequencing rather than optical approaches. And so here's a handful of lessons that I've picked up along the way. So um, one of the surprises was, as a scientist, you kind of think, how hard can it be to start a company? Just build a cell sorter, cash the check. What's the big deal? But we quickly learned that there's a lot of degrees of freedom and a lot of entropy in the process. And it really comes down to having cool people that are smart, ideally smarter and harder working than you. Those are the people you want to associate with, and find good mentors to help you avoid making the dumbest mistakes. You'll still make other mistakes, but it's just hard and it takes a long time. And then as a founder, fundraising is a full-time job on top of your full-time job. And fundraising, I don't think ever goes away. You're looking for money to fund your startup, you're looking for money to fund your venture fund. And then once you have a venture fund, you're looking for people to fund your portfolio companies and people to buy their products. So you're constantly chasing dollars. And you're also constantly building something that is worth that money. So that's a constant thing once you get involved in business. Or even if you stay in academia, you're still chasing dollars. You got to keep your students funded and fund your lab, and that's how it goes. Um, and as an investor, having been through the bumps along the way, I know that it's really valuable to have people who are very adaptive and flexible. They're motivated. I describe them as people who are willing to run through a wall, but smart enough to go around it. And they have they have kind of a good blend of being sort of unreasonable because they're so motivated to do this thing that nobody else is doing, but also have enough humility to know where they should ask for help. So it's a kind of a healthy blend of different characteristics there. And something I wish I understood better early on is that your colleagues are the people you go to college with. They're next door to you in your lab. They're going to be your colleagues forever. And it's really valuable to build those relationships early. And then you can call them up and they'll help you out and vice versa. And one of the things I did early on in the birth of phase two ventures was to write this book. Some of it was kind of how I approached making the review article I made for the spinal cord during my postdoc. When I got to my postdoc, I was feeling very insecure about all the stuff I didn't know. I knew there were motor neurons and a handful of transcription factors in the ventral cord, but the dorsal cord, I didn't know anything about it. And it was sensory-ish, mostly. And so I would take my newborn daughter in her bassinet and go to the coffee shop and, rock the bassinet with my foot and have a coffee and try to track down all this information. I started making all these notes, and then it kind of got to a critical mass where I'm like, well, this could be, maybe I could do a review article out of this. And I did. And so when it came time to do this fund, I thought, there's a lot of stuff I'm sure I'm not aware of in the venture world. I should probably take a minute and study as much as I can. And maybe one way to keep my attention to it and keep my nose in the book was to write it. And so this started off as just an exercise of trying to map out what I wish I knew as a founder early on about venture capital and what I want to know now as an investor. So this grew into 322 pages and 256,000 words of a survey of it, and I sprinkled in, some thoughts here and there that came from my personal experience. So here's that map I made during my postdoc of how to build a spinal cord. It's kind of a transcription factors meets kind of the subway map view of how each cell goes through these various It's like they took various classes to become specialized in their role. And this is also on the cover of the book. So um, my key takeaway from writing this book is that if you understand your counterpart, like if you're a founder, if you understand what venture capitalists are looking for, or if you're a venture capitalist, you understand how scientists operate, you can be a better partner. Um, as my 16-year-old daughter said to me not too long ago, I said something to her and she didn't hear me. And she said, "What?" I said it again. She goes, "Oh, I didn't hear you." And my confusion turned into hostility, which I thought was hilarious and a kind of a true nature of humanity, right? If we're confused, we get irritated. So if you can avoid confusion about how people are carrying out their jobs as an operator of a company or carrying out their jobs as a venture capitalist, you can hopefully avoid getting crossed up. And then I thought this is in the book, and I thought this kind of summarized how people get into venture capital, or maybe how I got into it. Um, you got all the kids and their various science projects, and this kid's the one who's trying to monetize it, right? Um, and then where are we in the current environment with venture capital? So when things work well, there's a virtuous cycle. You can start anywhere in this cycle, but let's start here with limited partners. So these are um, individuals with quite a bit of money, or something like the retirement fund for California, or um, Fidelity Investments. Somebody's got a boatload of money. They're going to put a tiny proportion of that towards alternative investments, meaning not stocks and bonds. They're going to go into real estate or venture capital or other things. They're going to give money to the venture capitalists. The venture capitalists are going to ideally have some sort of skill and insight to pick the best startups, help them build value, and then they will be they will go public or be acquired, and that will allow money to flow back to the limited partners, and the venture capitalists usually take a 20% split on that money. So there's a 2 and 20 model where venture capitalists get paid 2% a year to manage the fund, and then 20% of any profits that are returned to their investors. Um, and so I guess I just explained all this. We can look at it in print as well. And then in the current situation, um, things are a little bit sideways, especially in the life sciences, because of the overshoot during COVID, where we all got excited that Moderna is going to cure everything with vaccines, to maybe not, and we have to get FDA approvals after all. And so now there are fewer IPOs and acquisitions. So there have been over the last 2 or 4 years. It's always, you know, next summer it's going to get better, and maybe this is that next summer finally. And because of that, whoops, um, the LPs haven't been paid back. And so they're not very interested in investing any more money because they haven't gotten their previous investments back. So that means the venture capitalists don't have more money, or maybe they have less money than I thought they would. And so a lot of time is, or a lot of money is spent keeping the current startups going. So you've invested more than you wanted to in that, just to keep the lights on. And startups are struggling to raise. So good ideas are not getting funded. People are getting discouraged. It's just sort of the whole system's just kind of locked up. That being said, the best ideas kind of always get funded. Um, there's also a little bit of a draw from life science investing over to AI is going crazy right now. And we'll see if that how that plays out. There's a good chance we'll have a bubble there as well. And then biotechs have a kind of valley of death at multiple points. And so, you're here and you need to get over here. You need to get your financing. So there's kind of a valley of death getting your first money. There's valleys of death to get to acquisitions or IPOs or FDA approvals or getting your machine built or getting it through electrical emissions testing. There's all sorts of hiccups here. And so ideally, your venture capital partners will help you build a bridge and get you across that, right? And fundraising, there's kind of the PR version of it, and then what does that look like underneath? So there'll be a press release that says we raised $70 million, right? Great. But it's not like they show up with the suitcase full of cash and unlock it, and here you go, here's all the cash. Oftentimes they'll do what's called tranching, right? So here's of that $70 million, I'm going to give you $10 million to get to phase one approval, say, or phase one build or whatever, this chunk of your project. And then if that goes well, I'll give you the next 20 million to get to the next chunk of your project. And in pharma, you think about FDA approvals. And then if you make it to phase three, I'll give you the rest of the money. And so um, the operators are sweating it to get to phase one. They're sweating it to get to phase two. They're sweating to get to phase three. And ideally, they're communicating with the venture people to keep them apprised of what the progress is, what their challenges are. Do they need more help? Is there an exception? Can we bring the money forward a little bit? And the operators need to tell all their vendors, here's the map we're on. I need to get from A to B in order to unlock this next round of funding so I can hire you for the next work. So there's a lot of communication and diplomacy that's required along these paths. And there's kind of valley of death fallouts along the way where you have failure rates that just kind of stay steadily out there. So some of the key takeaways overall are that from my perspective, it started with the science, and then I tried to learn the language of business early on. And I'd say if people who are coming from a banking background or a finance background, they'll have to flip the script there and learn the jargon of their counterparts. Build relationships early before you need them. Like if you are raising money, go talk to people 6 to 9 months ahead of when you need that money and just say, here's what I'm up to, just letting you know where I'm at. I would love to work with you in the future. And then they see, okay, here's a person who took the time to communicate with me. I see that they're at point A. Later on, they sent me an update from their newsletter that they're now at point B. And now when they call me and ask for money, I can see they're almost at point C. So you can see traction. You don't just come out of the woodwork. You've done the work to get to know people. And then understand that failure is constant. Whether you missed your exit on the freeway or you stubbed your toe, there's just constantly going to make hiccups. The FedEx goes to the wrong dock, or it got thawed got held up in customs. That person went on vacation. You did the wrong size. It's just constant friction and mistakes. And so, being um, graceful as you can when that happens, and being gracious to others when they make the mistakes, that's important. You want to not make mistakes, and you want to help people not make mistakes. But if you're just yelling at people all the time, that's not going to help them stay motivated. And it's not a linear process, right? There's this, kind of famous cartoon or meme of, success is up and to the right. And that's probably what people portray that on their LinkedIn profiles or their Instagram reels, but we all know that it's a mess trying to pull this all together. And, if you think it can be any aspect of your life, but it's going to be messy. And I think that's what I had to share today. I'm happy to stay in touch with you guys. You can reach out to me on LinkedIn if you want to stay in touch. And if you want to get my book, you still have a few shopping days before Christmas for all of your family members. And it's on Amazon in hardcover, softcover, and Kindle. So I'm happy to open up and take any questions. Uh, Himani, you want to go?
Yes, I can go ahead. First of all, thank you for the session. It was very interesting. I truly agree to the point, like all you said, science people need to learn the business language and business people need to learn the science language. That was like very interesting. I have a question around like valuation of a company. Like, how do someone like a person with science background, they come up with the science, cool science, and then how, like, who are the people that help them with, and like we do hear the terms angel investors and even funding, like dilutive funding and non-dilutive funding, like at what stage these steps are done and like how these are done, and is there like, I know you like venture capitalists are happy to help us in that phases, but when to reach them, is it like certain point of your product that you should reach to them, or like what are your takes on it?
Yeah, so there's a few questions in there. One is, let's start with when do you have kind of a critical mass, I guess? Is there something here that can be a company or the beginnings of something, right? Oftentimes you'll have something that's high enough quality, you've published it. So you've gotten some peer review. There seems to
Mm-hmm.
be a good story there. Um, and it's important that you have talked to your tech transfer office before that to make sure you filed something. That's a classic rookie mistake is to publish first, and that will make it very hard to patent, maybe impossible. Um, as far as the value and when people are interested, that's a judgment call. It's very case by case. I would say there are a few factors. One is, do you have a team that is ready to work on it?
Mm-hmm.
It can be you as a freshly minted postdoc or grad student. You can have the support of your advisor if they're cool. That can be great. Um, or can you find people in industry who are ready to mentor you or join your team, as an interim role, perhaps? And then how ready is your technology? Can you build a prototype of it? Do you have
Mm-hmm.
a, kind of a toy version, proof of principle version of it? And then it really depends on how complex that problem is. If you're building a cell sorter, can it sort cells? Can you show that it can do that in any way? If you're making a drug, do you have animal data that shows it does what you think it's going to do? Um, and then there's kind of the timing of what you're building
Mm-hmm.
and what the world is ready for, right? I recently I've been playing Scrabble online with my sister, and it's like you can have a great word, but if there's nowhere to put it on the board, you can't play, right? And so the world has to be ready for your offering and value it. And sometimes, Steve Jobs would say, they don't know what they want. And, he's maybe we have survival bias for that guy. But usually you can find potential customers and talk to them and see if what you're doing makes sense to them.
Okay.
And then if I don't know if you're in where you're located, but oftentimes there's entrepreneur clubs or business clubs or business, startup incubators and environments to kind of plug into and just get started talking
Okay.
to people.
uh, okay. Thank you so much.
Sure. Helen, do you want to ask a
Yes.
question?
Hi, thank you so much. Thank you so much, first of all, for Berkeley Venture to organize this event. I am an incoming student of MTM program at Berkeley, and I'm in the process of starting a company with my partner, but we have very early ideas that as you mentioned, that we haven't published yet, but it is probably not asset yet. So I'm just wondering, so today's market, do you prefer a company that build a broad platform technology or those that are focused on, how do I say, like a de-risking one lead asset
Yeah.
with possibly a quicker clinical trial down the line? Thank you so much.
Sure. I would say a lot of fundraising is person to person interactions. So as much as so you could find that some venture funds are like, we want platforms, and they're out there looking for platforms, right? And there's others that we want assets. We want one thing that's all the way down the line. And so I think it comes down to finding that's one cut is you can kind of figure out is that whole firm interested in a certain approach? And then ultimately, you're going to want to interface with a person at that firm, and they're going to get personally excited about it, and they're going to advocate for you and want this to be their deal. So, um, as crass as it sounds, it's not unlike dating, right? You find somebody that's cool, you both see a twinkle in each other's eye, and then you start building a relationship, right? Um, and so it does come down to those personal relationships at between your company and that company. And then it's case by case from there. It really depends on who's sitting at the table.
Oh, thank you. Matter of fact, can I just follow up another question?
Sure, sure.
Yes, talking about person to person, I'm very curious, like us, because I've been, attending meetings about like pitch competition and stuff like that, and I realized that a lot of them has very early on ideas which are fascinating, and it sounds like it could be anything, could be a blockbuster in a few years. But other than that, what else are the VCs are looking for? Like maybe the partners, how their background, or because
Yeah.
as I said, a lot of times it's just a dream.
Yeah. And the best drug hunters still have a hard time getting drugs to work, right? It's complicated. Maybe one way to make yourself um, ready to be seen by a venture group is to think about what does your data room look like? Do you have a data room? What's in it? So that exercise can help you begin to gather the materials you need to support explaining your company and where you're at. Also, writing a grant proposal can be another way, or writing a paper. You know, those are good exercises to get your ideas into a tractable form with goals and an outcome.
Van Loan, do you want to ask your question?
Yes, thank you, Tiffany. Hi, Will. So you said earlier that even with the hard times that we're in, the best ideas still get funded in biotech. I just wanted to see if you have specific ideas about how you define best ideas, just in terms of the area where phase two VC has been focusing on.
Um, it's always case by case, but you kind of know it when you see it. So it'll be a team that has maybe done something before. They have a track record of innovation. They have a well thought out idea. They have preliminary data that's maybe validated by a third party. That's another cool thing to get the attention of venture, where it's not just my experiments, but I've had the experiment has been replicated by somebody else. Um, and so it's the team, the technology, and the timing. It keeps coming back to that, right? So the technology has to work. There has to be something real there. The team needs to have the wherewithal to understand what they have and to advance it to the next stage, and hopefully have enough humility to ask for help if they if they foresee it growing beyond their skills or faster than they can gain those skills. And then the world needs to think that's cool. So if you have a way to show me that here's how all drugs bind to all proteins, you know, we go sell it to AlphaFold, right? Um, that would be cool. And AI money would pay for it too, right? That would be kind of the sweet spot right now, I think, for finding funding. So if you have that, give me a call.
Matthew, you can go ahead.
Thank you. Great presentation, Will. And I played 14 years jazz, so I do believe in the adaptability of doing music. I think
Right,
there's
right.
a lot there. I had a question because we have been talking to a number of venture and angels, and the thesis that the venture is based on comes up again and again.
Yeah.
And, they provide a lot of networking, they provide a lot of business support and experience. But your uniqueness being a scientist, laboratory scientist, is really valuable to me. But I was hoping you could explain, how does that come to play in the boardroom that we might see differently than these other venture?
Yeah. I think there's kind of, broadly speaking, two categories of venture folks, right? People who come from a scientific background and then are not as well versed in the finance, and the people who come in the finance approach and they're not as well versed on the science. And ideally, you have a blend of both. There's going to be good reasons to have people who are really good with the money to go get you, a debt instrument at the right rates from the right people, and people who have the science to make sure that you're building reliable data to support your mission, right? Um, and then as far as the the theme or thesis of a venture group, I think that's often helps you know, like if you're building a box, you don't want to talk to a company that only does pharmaceuticals or only does, medical software. Um, but if you can kind of get to the right broad strokes, it's going to come down to, even if they have a thesis, they're often willing to flex that if they're excited about your project. Hope that helps. Yeah. Christine, you want to go?
Hi, I'm coming from more of a very solid life science background and almost a VC phobic, just thinking and like, oh, how could it possibly get into this area? Um, so I think I'm hearing a lot of messaging or things coming back to this idea of building the right team being essential.
Yeah.
And I think one of the reasons I've had shied away from the idea of VC in the past, like even particularly like having done an HHMI fellowship in translational medicine, like that almost made me more phobic. From like hearing this sort of narrative that the MBAs or the suits are going to interfere with this, take control of this, and run off with it. But I was interested to hear your story about one of your early startups getting an army of MBAs first and getting them to go out and um, get the ball rolling for you to put some things together. I want This is one of a couple of questions, but like, how did you approach managing that as someone like with the life science background?
Yeah.
How did that sort of training translate into being able to build and manage that team
Um,
where the
yeah.
science lead?
Yeah, it was hard. I started off at the beginning, and I think similar maybe to what you're saying, I was a little insecure about what I knew on the business side of things. And I think, you know, in your case, it sounds like, you got that HHMI fellowship, so you're probably going up and meeting fancier and fancier people, and so I found
Hmm.
that to be, it's like, oh man, there's more mountain up ahead of me the higher I go. Um, and then on the other side, if you just kind of hunker down and do good work and kind of take the time, you can make progress and you can learn about other fields. And ideally, one of the things that you get out of grad school is how to do the research and how to teach yourself, right? And
Hmm.
to some extent, it's some self-teaching. It's finding new peers that are going in a similar direction, right? It may not be that your mentor in academia is the right person because maybe they're
Yeah.
not business minded. So it's finding new peers to support you and, share your wins and losses and try to figure it out together, right? Um,
And Sorry, that kind of leads into my second question.
Sure.
Um, I certainly hear and kind of like regret not following that advice of um really building the relationships with your grad school colleagues, your mentors early on in your in your career, with this kind of approach in mind, with these kind of goals in mind. And I think it's something I may have taken for granted, having been in a translational lab, uh, having my background was specifically in DNA vaccines.
Uh-huh.
And they kept running into this wall of the delivery device, the gene gun, being the hang up. And
Yeah.
you know, seeing those mistakes being made, but being at a very, very like junior level. And just not, and at least in my mind, I don't think I really took the bull by the horns in terms of trying to stay connected to people who might still be doing that work or left that work. Long story short, a lot of folks have sort of dispersed and gone into academia. That kind of my network is not really interested in the VC path. So
Yeah.
if you're someone who's kind of later in your career, but still like somewhat interested in, you know, circling back around to this, how do you have any advice with regard to how to jump into those sort of teams or start, rebuilding like a sort of
Yeah.
network that
Well, yeah.
has that wisdom and success?
One way is you could look for people who are publishing in that area. If they have academia and industrial members of those teams, those might be people that are in that zone. Are there companies that
Mhm.
have been formed in that area? Are there early stage companies that are just barely getting going? NIH grant winners, you can look on NIH Reporter to find who's going after it, or
Okay.
clinicaltrials.gov is another resource, or
Mhm.
Ask Claude. I don't know, there's all sorts of sheets on top of it now as well.
But maybe even like look outside the US at this point. I kind of I
Maybe.
wonder about, this in the regulatory environment, that we're
Hmm.
in. I'm assuming that is really affecting the cycle, the
Yeah,
slowing down that you described.
I would say
Um,
that if the technology works, the regulatory can follow, right? But
okay.
if there's a showstopper, like I don't know much about the gun you're talking about, but if there's something
Yeah.
that kind of a holdup in the whole field, like LNPs, you need LNPs to get your stuff into the system, and if those aren't good enough, then the whole field's kind of hung up on these technological inflection points, or the regulatory agencies are, or whatever. There can be some human factors about how we're thinking about the problem in addition to the biology and engineering.
Thank you.
Yeah, thanks for your question. G, do you want to go?
Oh yeah, thanks, Williams. So I have two questions. So I'm
Sure.
also the MTN student from UC Berkeley.
Yeah.
So the first question for me is like, how do you think about the AI will affect the field of the consultant or be like the advice this area? I mean, like for your company for the phase two venture company. So you say that you give advice for the people who want to start their startup. So I'm really curious about that. And for the second question is like, do you think that for the venture side, is there any preference for the venture capitalists to make a decision for the different technology like because I know the tech and deep tech is different. So is there any preference for them to put their money into this startup? And also like, is there any differences between the tech and deep tech for the funding pool for these two areas? So that's my question. Thank you.
Yeah, so I think there's something like 2,500 venture companies in the United States. So it's probably at least one of them will overlap on what you're interested in. So I was saying you know, in life sciences, you could break it into like therapeutics, med device, software, and tools and diagnostics, right? And then within each of those, there's got to be a person at that firm who's really psyched about, your deep tech question. And so you could almost search LinkedIn for venture and whatever your keyword is, and maybe you'll find that person that is psyched about what you're interested in. And you can ask them, does this idea seem good? Um, if not, do you know somebody else I can talk to? It's there's a lot of the times it's just kind of doing the work and slogging it out, and it's not something I don't recommend doing an email campaign to spam everybody. I think it's really person by person, building a network, being a reliable counterpart to that person, um, being a resource to them as well. If you're serious about this, they're going to want to know you and what you're working on and who you're talking to, right? So it can be a two-way street of helping to advance this problem and build a network that way. And then I forgot your first question.
Oh yeah, my first question is like, how about the AI things? Do you think that AI will affect a lot about the consulting area? I mean, for the like, give some company for their to give device.
Yeah, yeah, AI is going to do things that start with English first or language first, right? Since the LLMs are best suited to that. And then I think, um, and if you're having it help you with a regulatory document, you can read it and see if it looks reasonable, right? On the other hand, and so I think that will go probably pretty quickly. The next level is, can I use AI to analyze proteins and nucleic acids and metabolites, which humans can't really look at and make sense of, right? It's kind of squiggly lines. And, you know, I don't know. You look at a UMAP projection, it's like, well, okay, there's fuzzball one, fuzzball two. I guess that's good, right? It's not like reading a paragraph of text. So if you're using AI to help you generate those data, it's going to be hard to tell if it's giving you reliable data until you have a wet lab validation of that. And so I think um, AI models are probably in need of vast amounts of wet lab data that we're probably not thinking about in the right way. It might be kind of crappy data, but there's so much of it that it works out from an AI perspective. Like if you're building an LLM and you scraped Reddit, I'm not sure Reddit is the best form of literature in the world, but it works, right? Or all the Gmail emails, right? It's enough kind of crappy information that it works. And so thinking about wet lab experiments from a kind of a broad but shallow approach might be interesting.
Okay, thank you.
I kind of went
Okay.
on a rant there, sorry.
Thank you so much, Will. We are at the top of the hour. Really appreciate you taking the time to talk with us and to share your journey from being a scientist to running a venture firm. And we really do appreciate you talking with us today. Thank you so
Yeah,
much.
thanks. Thanks, everybody. I really appreciate you guys inviting me. And if you have follow-up questions, just ping me on LinkedIn and happy to stay in touch.
Thank you so much.
All righty. All right. Talk soon. Bye-bye.